Welcome back. We are now doing chapter 28, and there's only going to be one slide that I will present, and that's going to be included in your last exam or your final exam next week. And uh, that's for chapter 28. I'm not going to be requiring you to study part two. So, whatever you see in the slide is going to be what you're going to get in your last chapter. Not really last chapter, but second to the last chapter of human anatomy and physiology. So, we finally got into pregnancy and development. And here are some words that you might need to go and look up. Conceptus means developing offspring. Pregnancy is a state of carrying a developing conceptus. Gestation period is a time from the last menstrual period until birth, which is roughly about 280 days. An embryo is the conceptus from fertilization up to the eighth week, and a fetus is the conceptus from the week 9 all the way to when the time that a baby is born. And here's a good picture, diagram from fertilization all the way to the 12th week, and possibly the sizes. So what happens, guys? First of all, we know that before fertilization can occur, the sperm must reach the secondary oocyte, which is actually what has been ovulated. A secondary oocyte is the ovulated egg, we call it. And the oocyte is viable from 12 to 24 hours. After that, it cannot stay on a suspended animation under metaphase 2 without being fertilized. After 24 hours, it will not be viable anymore. The sperm, on the other hand, is viable from 24 all the way up to 48 hours after ejaculation. So, for fertilization to occur, coitus must occur no more than 2 days before and at least 24 hours after ovulation. And fertilization is the process. It's actually where the sperm's chromosomes will combine with those or with that of the secondary oocyte to form a fertilized egg, which we call a zygote. So most ejaculated sperm do not make pretty much like the entire trip to join the egg. Some of them will leak out of the vagina immediately after deposition. Some are destroyed by the acidic environment of the vagina. Some may fail to make it through the cervix. Some are dispersed in the uterine cavity or destroyed by the phagocytes. And only a few thousand, if not hundreds, out of the million can actually or will actually reach the uterine tube. So the sperm must be capacitated before they can penetrate the oocyte. That means their motility must be enhanced, and cell membrane must become fragile enough to release the hydrolytic enzyme. See, they have what you call an acrosomal cap, and this particular acrosomal cap, the one in blue right there, must be fragile enough in order that it breaks easily to release the hydrolytic enzyme that's going to digest the path by which they can use to penetrate the egg. So secretions of the female tract actually help and we can thin out this acrosomal membrane. So the word capacitation is a process that we use pertaining to the things that will happen to the sperm so that it will have the capacity to fertilize an egg. It occurs while migrating through the female reproductive tract. The process in the sperm involves, some cholesterol withdrawal, surface protein redistributed, some calcium influx, pretty much which will increase motility and whiplashing of the flagellum. A capacitated sperm can penetrate the corona radiata, contact the zona pellucida, which is actually the protein that surrounds the egg, and undergo the acrosomal reaction. So let's take a look at the acrosomal reaction and sperm penetration. First of all, we look at the approach. The approach is aided by enzymes. The sperm will be weaving right through, okay, weaving right through the corona radiata. Let me take a look at my laser pointer right here. It weaves right through the corona radiata, and this is actually your only a small portion of that area right there of the egg. You can see how small the sperm are and how big, humongous this secondary oocyte is. So it approaches the egg, or they will approach the egg. They are aided by enzymes, and this is actually a coronal radiata. Now the hyaluronidase, which is actually the enzyme on the cell surface of the sperm, acts to digest connection between the granulosa cells, causing them to separate. Well, guys, unlike in the picture where you have seen really like hundreds of sperm and only the and they're all competing to get into the egg, that is not really the case in a real scenario. They are not really in competition with each other. All of them are actually aiming to digest the pathway by which they could get into the egg. A lot of them will die. A lot of the sperm will already die and would have already used their energy before they can even digest the entire path. So in reality, they are actually doing this to help each other out. The first of the sperms, the strongest, and the first of the sperm that go there will never reach the egg because they will die of exhaustion because of what they're doing to digest the path, and they're not going to be able to reach the egg. And therefore, those who are behind them, okay, will continue the work on digesting and digging in. This is just like digging a tunnel. So all of them is actually going to do whatever they can do until they all drop from exhaustion, and they're not going to be able to make it anymore. But what's going to be making it will be the one behind it, and eventually the last of them that could actually penetrate the egg without those granulosa cells that have already been like wiped away or digested, and that's going to be the luckiest ones. All right, so this is a concerted effort of thousands or millions of sperm in order that they can approach the egg. So the sperm will penetrate the egg with the help of the enzymes that are actually on their cap. And they will be digesting the connection between granulosa cells, causing them to separate. You can see here that the granulosa cells are separating, and then they are actually going to fall off. Sperm heads then bind to the sperm binding receptors in the zona pellucida. This is actually your receptors right there. These are the receptors. This is the zona pellucida. They have to digest the zona pellucida in order to get into the receptors. Now, as they bind into the receptor, it causes the sperm membrane calcium channel to open. Once the calcium flows into each sperm, it triggers what you call the acrosomal reaction. The acrosomal reaction now, okay, when triggered by the calcium influx, pretty much enzymes from many sperm are released. Then they will be digesting the holes in zona pellucida. You can see here that they're starting to digest a hole right through the zona pellucida. These enzymes would include the hyaluronidase, the acrosine proteases, and others. Hundreds of acrosomes in the region all release enzyme at the same time to digest the zona pellucida. And many are needed to clear a path to the oocyte membrane. So after the path has been cleared, now you are in the binding right there. After the path has been cleared, a single sperm will forcibly swim towards the oocyte membrane. The acrosomal color on the rear portion of the membrane binds to the oocyte and plasma membrane and sperm binding receptor. The binding then will cause the oocyte to form microvilli that wraps around the head of the sperm. These trigger fusing of oocyte and sperm membrane. So immediately there's going to be like, okay, you're the first one to come in, and then they will be embracing each other and says, okay, welcome and come in. But only the pronuclei of the sperm gets in, not the rest of the sperm. The what do you call this? The mitochondria is left outside, and the tail is left outside. So the oocyte and the sperm membrane will now fuse. The cytoplasmic content of the sperm will now enter the oocyte. Okay, and the 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 sperm cell membrane and the mitochondria are left behind on the oocyte cell membrane surface. It will not be entering the egg itself. So, what is our guarantee that there's only going to be one sperm that gets inside? What if, on different sides of the egg, there's going to be many, many sperm? And of course, that is really what's happening. Would there be a polyspermy? Well, in humans, polyspermy do not occur, okay? It's only monospermy, simply because there is actually a block to polyspermy. We have actually what you call a one sperm per oocyte condition. Now there are two mechanisms that would ensure monospermy. The oocyte membrane block. This means that when a sperm binds to the sperm binding receptor on the oocyte, it causes the oocyte to shed all other sperm binding receptors. Other sperm can no longer bind to the oocyte plasma membrane because of this. The second one is the action of the zona pellucida. Okay, the zona reaction, also called the slow block to polyspermy, is actually what happens when the entry of the sperm into the oocyte triggers a calcium surge from the oocyte endoplasmic reticulum that causes the activation of the oocyte to prepare for a second meiotic division. Now, this is actually a cortical reaction. The granules located just inside the oocyte plasma membrane would release a zonal inhibiting protein, or ZIP, into the extracellular space below the zona pellucida. This ZIP will destroy the zona pellucida sperm binding receptors. So all of these receptors, immediately after one sperm docks into the receptor, all other receptors are going to be dismantled. Okay? And therefore, no other sperm can actually come in anymore. So what happens if there's going to be really a simultaneous another sperm? Well, guys, that particular fertilization is going to be null and void. It's not going to be possible to have a triploidy. There's not going to be two male pronuclei that's able to penetrate one female pronuclei, not in a human. Okay, we do not have any triploidy. Therefore, that particular fertilization is not going to become a baby. All right, it will not involve any development. So now we are going to be in the completion of meiosis 2. After your sperm deposits the pronuclei, there's now the completion of meiosis 2. Finally, your egg that has been a secondary oocyte that has been in suspended animation in metaphase 2 will now have some changes that's going to move it into continuing and finishing up from metaphase 2 all the way to the end of meiosis. It involves a calcium surge that triggers the completion of meiosis 2 in oocyte, resulting in an ovum dividing it, and with two daughter cells, one becomes the ovum and the other one becomes the polar body. The ovum nuclei swells to become the female pronuclei, and the sperm nucleus moves towards the oocyte nucleus. Okay, it also swells. to form the male pronuclei. There you go. This is actually how it would look like. That's going to be your male pronuclei and your female pronuclei. After the sperm penetrates the secondary oocyte, the oocyte completes the meiosis II, forming the ovum and the second polar body. Okay, and right here, when it divides, actually this is not the male pronuclei. That is the second polar. That is the first polar body. This is actually this is already the second polar body, and this right here is actually the sperm pronuclei. This is the sperm pronuclei, the female pronuclei that has just divided. This is the polar body that is actually one of the daughter cells that as a result of its division. Now your sperm, okay, is the male pronuclei, and the female pronuclei would both swell, forming male and female pronuclei. They will stand next to each other. Okay, the DNA in its pronuclei would replicate. The pronuclei approaches each other, and a mitotic spindle forms between them, as if really doing mitosis. That's going to be where they're going to be ready for the first cleavage division. The chromosomes of the pronuclei would intermix. At this point, fertilization is accomplished, and the cell is now called a zygote and is ready for the first cleavage division. So, from the zygote, this is actually your zygote, to your blastocyst. This is now your blastocyst. The embryonic development would continue as the embryo travels through the uterine tube into the uterus, while it floats freely until it finally implants. And there are significant events in this process, and we'll divide it into two parts: the cleavage and the blastocyst formation. These will be your cleavages, and then this is now your blastocyst that has formed. Let's go through them and figure out how your blastocyst is formed. First, in the cleavage, it occurs while the zygote really moves towards the uterus. There's actually rapid mitotic division of the zygote. It produces cells with high surface to volume ratio that enhances the uptake of nutrients and oxygen and quick disposal of waste. The first cleavage would occur after about 36 hours and produces two daughter cells called blastomeres. You don't have the blastomere showing here, which will continue. This is already the continuation of the division of blastomeres. Okay, the first blastomere or the 2-cell stage is not shown here, but you have already the 4-cell stage, and possibly this is already like a 64-cell stage. After 72 hours, a cluster of cells containing 16 or more cells is now referred to as the morula. This is now your morula. So around 4 to 5 days, where the embryo now is about 100 cells or more, is now referred to as the blastocyst. It is now going to reach the uterus. It's right here now. It's hanging in there, okay? Blastocyst is a fluid-filled hollow sphere that is made up of trophoblast cells, and the trophoblast cells will be this surrounding, okay? The surrounding cells here, okay? And the inner cell mass right there, which is destined to become the embryo, the part of the embryo. They display immunosuppressive factor, and they participate in the placenta formation. Those trophoblast cells, those single layer of cells surrounding it, okay? The embryoblast is actually your inner cell mass. This is actually the inner cell mass. A cluster of about 20 to 30 rounded cells. They become the embryonic disc, which will form the embryo, and about 3 or 4 extra embryonic membrane will arise from there also. And the fourth extraembryonic membrane, which is called the chorion, is formed by these trophoblasts. That's actually the fourth extraembryonic membrane. So you have actually four extraembryonic membrane, and three of them arises from the inner cell mass, and then the fourth, which is euchorion, arises right there from the trophoblast. So the blastocyst floats for about 2 to 3 days and is nourished by uterine secretion. And then between the 6th and the 7th days, it will now be implanting. Now, how does it implant? Well, remember, guys, that this is now a living organism and it is trying to survive. It wants to hang on to the wall of the womb that it's actually next to. And not only to hang on, it wants to get in and be secured right in the womb. So just like anything else, we could regard this really like the action of a desperate, temporary, like living organism that wants to like feed on to another living organism. Call it a symbiotic, a parasitic symbiotic relationship. But that is actually too much. We don't want to refer to our babies as parasites, but in the beginning, that's exactly what their initial not really thinking, but their initial reactions are just like a parasite. They want to grab on to the source of food and stay right there until they're ready to be by themselves. Of course, they're not going to be permanently staying there. They are only going to be temporarily staying there while they are actually forming. But they are feeding on the mother, and therefore you could pretty much call it really like a parasitic involvement right there. So, the trophoblast, okay, this is actually the outer portion right there. The trophoblast cells would adhere to the site with proper receptors and chemical signals, and then they will create what you call an inflammatory-like response in the endometrium. So when there is actually an inflammatory-like response, it will be felt by the mother. The mother will be inflamed. Okay, this portion here, which is actually the side of the mother, will receive the inflammatory chemical from being released by the embryo and will swell up and will feel like it is actually like inflamed. And when there is inflammation, there should be healing. Am I correct? So the two of them now is going to be like touching each other. And let me show you the next part. Right there you go. Now they are actually touching each other right there. And your trophoblast cells will continue to secrete inflammatory chemicals that's going to be picked up by the mother. The mother's side right here is going to be receiving a lot of inflammatory chemical, and it will be inflamed. Okay, it will be inflamed, and it will degrade. It will actually be degraded by the chemicals. Okay, so at this point, there are two types of trophoblast cells that would proliferate and take over in digging in. I call it digging in, so that the entire embryo could actually get inside the wall or get into the wall. The cytotrophoblast, which is actually the inner layer of cells, and the syncytiotrophoblast. And these are actually the outer layer of cells. They will lose the plasma membrane and become multinuclear mass. This is your syncytiotrophoblast, this yellow stuff right here. And the cytotrophoblast will be this part here. The syncytiotrophoblast will continue to secrete inflammatory chemicals and will help in digging in, okay, if that's a proper word, so that the embryo can actually penetrate the wall itself. Okay, as it is actually already getting inside, the mother now will start to heal up, because after an inflammatory response, healing will need to occur. But in the healing part, what the mother side is going to do is that it's going to form a layer. Okay, it's just like it's embracing, taking in the embryo, and it is not sensing. The embryo is actually secreting a chemical that is going to render the macrophages and the white blood cells of the mother blind to the presence of a foreign body, because that is a foreign body. Okay. And right here you have a fully implanted blastocyst. And day 11, the blastocyst has already been fully implanted. And right here, you already have the inner cell mass forming what you call the embryonic disc. The embryonic disc will start as a bilayered disc. Okay, the bilayered disc forms what you call the epiblast and the hypoblast. There you go. That's on day 11. So in cases where implantation failed to occur, the uterus becomes non-receptive again. About two-thirds of all the zygotes that are formed would fail to implant by the end of the first week or would just spontaneously abort. And an estimated 30% of implanted embryo later will miscarry because of some genetic defects of the embryo or uterine malformation or any other unknown problems. Implantation is usually completed by day 12 after ovulation, and about the same time the menstruation would occur. So if you miss your menstruation already, you are already like possibly on the 26th day of the menstrual cycle or more than that, your 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 baby would have already implanted. If you missed your period now, and you're already pregnant because your baby had already implanted right there in the uterus. So the corpus luteum at this point is maintained by the human chorionic gonadotropin to prevent menstruation. And right here is just a good graph showing us the different types of hormones that are present during pregnancy. Okay, the human chorionic gonadotropin is secreted by trophoblast cells and later the chorion. It prompts, this is actually a human chorionic gonadotropin, the orange one. Okay, and the first 2 weeks of pregnancy, it will spike. Okay, it will prompt the corpus luteum to continue secretion of progesterone and estrogen. It promotes placental development via its autocrine growth factor activity. And pretty much the levels will rise until the end of the second month. And therefore, when you are doing your pregnancy test, you are actually checking for the presence of the human chorionic gonadotropin, which really a real pregnant female would have. If you just missed your period and you are not really pregnant, or you just you just having one of those months where your ovulation is late and everything, or you're probably under stress and you missed your period, you think you're pregnant, you are not. If you do not have an increased human chorionic gonadotropin, because only pregnant females will have an increase really of human chorionic gonadotropin. It declines though, okay, as the placenta begins to secrete progesterone and estrogen. You can see progesterone and estrogen level will now go up, but pretty much low values occur at 4 months, but it will continue the low values all the way to the rest of pregnancy. So the placenta is formed. It's actually a temporary organ that originates from both the embryonic and maternal tissues. The embryonic portion of the placenta would include the inner cell mass. This is actually inner cell mass, which gives rise to the layer of the embryonic mesoderm that lies near the inner surface of the trophoblast. And together, these structures form the chorion that develops finger-like projections called chorionic. This is actually this will form the chorionic villi. So now the chorionic villi are then now invaded by new blood cells. So you have the blood cells invading the chorionic villi. This is actually your chorionic villi right here. And with continuing erosion of the endometrium produces large blood-filled lacuna and the stratum functionalis, okay? And the villi lie in the intervillous spaces that is totally immersed in maternal blood. So this is actually your chorionic villi right in this section right here, okay? This is your syncytial trophoblast continuing to work on forming, uh, on eroding, on helping in eroding the maternal part so that that will give more space uh, for the implanted embryo to grow. And here you have uh, more erosion showing, but more of the maternal blood and more intervillous space containing the maternal blood is actually developing. Okay, but right here in the inner cell mass, you could see now that you have already the formation of the from the bilayer. Now you have the three layers. You now have your ectoderm. You have a middle layer, which is the mesoderm, and you have the innermost layer, which is called the endoderm. So now the placental portion of the maternal portion of the placenta will include the decidua basalis. The decidua basalis is this section right here. Okay, that's the decidua basalis. And the stratum functionalis of the endometrium that's located between chorionic villi and the stratum basalis of the endometrium. The decidua capsularis, okay, this decidua capsularis is again part of the maternal part, okay, provides portion of the placenta that expands to accommodate the growing fetus. And the villi in the decidua capsularis will degenerate as the fetus will grow, while the villi in the basalis will increase in number, right here, will increase in number and branches. Now together, the chorionic villi and the decidua capsularis will make up the placenta. So now the placenta is fully formed and functional by the end of the 3 months. It provides nutritive, respiratory, excretory, and endocrine functions. Maternal and embryonic blood supplies normally do not intermix, okay, but some fetal cells may enter maternal circulation. What happens here is that any food that the fetus will derive from the mother goes by way of diffusion. It's not because the mother's blood is mixed into the fetus or the fetus blood is mixed into the mother. Remember that the blood types of a mother and a child sometimes may be different. They cannot really mix, okay, most especially if their blood types are different. So placental barriers would include membranes of the chorionic villi and the endothelium of your embryonic capillaries. You could see here, guys, that the maternal vein is really not mixing with the blood of the fetus. There's no intermixing of the blood. They exchange gas, okay, and nutrients by way of diffusion. So the placenta secretes the human chorionic gonadotrophin from the beginning until the production of estrogen and progesterone would increase. If placental hormones are inadequate, when your HCG levels will wane down, the endometrium degenerates and pregnancy is eventually aborted. So throughout the pregnancy, blood levels of estrogen and progesterone should increase, okay? And that would also be preparing the female when they are lactating. The mammary gland would develop at this point. The placenta also secretes placental human lactogen, okay, human chorionic thyrotropin, and relaxin. Now, during implantation, the blastocyst begins by being converted into gastrula. Whenever there is already gastrulation, that's already the beginning of the formation of the organs. The 3 germ layers as well as the extra embryonic membranes developed from the gastrula. The inner cell mass divides into two layers, the epiblast and the hypoblast, and I showed you already where the picture is. And pretty much that is actually what you call the embryonic disc. Now, I am now going to get you into the extra embryonic membranes. These are membranes that are outside of the embryo that are helping or assisting the embryo in its development. The amnion, for instance, is also the water bag. Okay, it provides buoyancy environment that protects the embryo. It helps maintain constant homeostatic temperature. It allows the freedom of movement. It prevents parts from fusing together. Initially, the amniotic fluid comes from the maternal blood, but later, fetal urine contributes also to the volume of the amniotic fluid. The yolk sac. The yolk sac hangs from the ventral surface of the embryo. It forms part of the digestive tube at the later part, and during the development, it disappears because unlike the egg itself, for instance, chicken egg, the yolk would persist all the way to the end of the development because eggs would carry the chick far away from the mother, and nothing is going to be feeding the egg had it not been for the yolk sac. But in our case, our baby stays inside our body, and therefore we do not need the yolk sac. So the yolk sac could deteriorate and diminish, but it will not totally disappear because at the end of what do you call this? At the end of a pregnancy, or rather in the development itself, it may not be providing fully uh, food all the way to the end because we get our food from our mothers. It becomes part of the digestive tube. It is besides the fact that it is the source of the earliest blood cells also and blood vessels. Then you have the allantois. The allantois are it's just a small outpocketing at the caudal end of the yolk sac. Pretty much in in birds and reptiles, the allantois is a an uh, extra embryonic membrane that becomes that receives the waste of the developing fetus or the developing animal. But in the case of humans, our waste is taken in by our mother. So the allantois pretty much is just a structural base for the umbilical cord and later on becomes part of the urinary bladder. The chorion plays big time. Is actually going to help form the placenta. It encloses the embryonic body and all other membranes. So pretty much you have now implanted your blastocyst. Okay, you can now see that you have your 12-day blastocyst right here. You have your bilayer. I am now repeating whatever it is. And then after that, in the 16-day embryo, you could now see that it is now a tri-layer. You now have your primary, what you call the primary germ layer after 4 days only. And then finally, in a 4.5-week embryo, you could see that you have already the formation of the decidua capsularis, the decidua basalis, the amnion and the yolk sac are already well formed. And then in a 13-week fetus, you could see the baby that is already almost like fully developed. So it's now honing on, improving on the development of its organs. So gastrulation occurs during week 3. It starts during week 3 when the embryonic disc, this is actually your embryonic disc, starts to form the 3-layered primary germ layers. The primary germ layers will be your ectoderm, your mesoderm, and the endoderm. And it begins with the appearance of what you call a primitive streak. Okay, and it's actually a raised dorsal groove that establishes the longitudinal axis of the embryo. So pretty much we are not going to go, I'm not going to get you into knowing the actual gastrulation process. I'm not going to hold you responsible in this. It's really nice just to look at it, guys. But there's just one thing that I would like to note close to the end, and this is this one here. I would like you to be able to understand where the or what happens to the three primary germ layers. What will be the fate of the three primary germ layers? For instance, your ectoderm right there. It is now destined to become the epidermis, the hair, the nails, the glands of the skin, the brain, the spinal cord, the neural crest. Okay. Then you have your endoderm, which is actually the one that is at the bottom right there, the yellow one. It becomes the epithelial lining and the glands of the digestive tract. And then right there in the middle, which is actually your mesoderm, all others will actually arise from the mesoderm. From your notochord, which will form the intervertebral disc, the somite, which will be your the vertebrae, the ribs, the dermatome, myotome, the trunk, the limb, the musculature, to the kidneys and the gonads, into the cover of the serous membranes, okay, the connective tissues, the wall of the digestive respiratory tract, the heart, the blood vessel, all of these arise, a great number arise from the mesoderm. This one I am requiring you to know. Your knowledge will only up to this point, your exam, and you are only going to be tested up to this point. And the last of our lecture will be on genetics. And that will be the end of today's lecture. Have a good day.